Good morning, everyone. This is Friday night on the West Coast of America, and I want to thank you for having me. My name is Dr. Jeffrey Gross. My practice is called Spine, and more recently, also ReCELLebrate, which is the regenerative side of the practice. And I'm happy to talk to you about the cutting edge um, of regenerative medicine for the spine. So, hopefully, you can see our slides here, and we'll get started. We all know what inflammation is. This is for years studied in the medical circles. Even basic medical students understand the swelling and redness of this type of thing. But cellular inflammation is something very different, for which we end up with the swelling and redness that you see here in this picture. And actually, cellular inflammation is measured by the amount of NF kappa beta in the cell, which is a gene transcription factor. It changes the cellular machinery to make inflammatory type activity, telling its neighboring cells to be inflamed and that sort of thing, which ends up with the clinical manifestations of swelling, redness, heat, that sort of thing. So we take the microscopic to end up with the macroscopic, but continued inflammation of a body part can be the root of a disease. And we're going to leverage that information to learn something about regenerative medicine and apply it to the spine. So when we're injured, when something's going wrong, acute inflammation is a healthy process. It helps us heal. It's a successful path of acute inflammation without a residual, without long-term chronic inflammation, because as we know, chronic inflammation is something very akin to most disease processes. So if we hurt our ankle and we had an acute inflammation and it swelled and it healed and it felt better, then that's a resolved process. If there's ongoing chronic cellular activity, cytokines, inflammatory cascade that go on a chronic basis, that leads to arthrosis and arthritis, which is the chronic disease state of what would otherwise be a brief healing process of the acute inflammation. Young people heal better than old people. And this is one of the processes, but something is going wrong when we're not young anymore, and we tend to have a more chronic inflammatory cascade. The same thing happens in the spine. We have inflammatory changes of the disc, the facet joints, the surrounding vertebra, and this is called the degenerated disc complex. Some people call this degenerative disc disease. Well, I suppose it could be, loosely called a disease because it is an ongoing bit of chronic inflammation, but itself probably is just not a specific disease process. And we see now, correlated to MRIs, the modic changes or the vertebral body marrow changes of what's going on in these discs in the spine that take on this stress. Here on the two side views of the MRI, or what we call the sagittal views, you can see the darkening of the vertebral bones above and below a disc. And this is a T2 sequence on the right showing a bright color. So this is a bone marrow reactivity. The cellular activity is angry and inflamed, and this can become a chronic process, which can lead to ongoing spinal pain and problems. So back to the microscopic. There are many pathways that have been found to be associated with inflammation of the cell. There's the mTOR, there are the heat shock proteins, and something is going wrong at the genetic level when we can't turn off the chronic inflammation process. And that can beget a very angry disc process and pain. So when I rethink about inflammation, whether it's for the spine or any other part, I believe that this is not just a swelling, but it's a reactive cellular behavior. And it's supposed to be leaving the least scar tissue and the least functional impairment. But the chronic ill effects of chronic inflammatory change or chronic cellular behavior in this inflammation cascade is a problem. Think about how a young person heals to a scratched knee. How a 3-year-old scratches a knee and a few days later it's gone. But where an 80-year-old falls down, has a bruise and a scratch taking weeks to heal. Something is going on at the cellular level that's not allowing that healing in the older person to be as robust. That's where regenerative medicine comes in, and we've learned a lot about that. So again, if I can't say enough that the phrase degenerative disc disease is not a good one. This is probably better stated to be an acute and repetitive, superimposed, inflammatory and reactive sclerotic bone issue of the marrow surrounding the discs. It is a vicious cycle. Because what happens is as more inflammation affects on a chronic basis the disc end plate anatomic relationship, the structure then cannot take the stress as much, and the whole process gets worse and worse. As you know, the disc is made up of the nucleus in the center. This is the squishy part, the viscoelastic part. But the spine is really involved with pressure from the end plates above and below. The disc is rather acellular. The nucleus pulposus is mostly water. It contains collagen and proteoglycans and various proteins that, when they aggregate, are known to have a viscoelastic or a viscous or squishy property. It bonds the hyaluronic acid, which is often used in orthopedic settings. Very low density cells, as I mentioned, although while sparse, the cells do produce what's called extracellular matrix. The annulus fibrosus, or the tough fibers around the disc, are mostly collagen. And these are directed around and to hold and contain the nucleus, so the nucleus, when it's stressed downward and in different positions, it is not displaced. Of course, if it becomes displaced, then you have the classic slipped disc. Other structures in the body are made up with similar collagens, and such as tendons and certain vascular organs and what have you. Forget the disc for just a moment and let's talk about the bone. Let's talk about the vertebra. The end plate is the surface interacting with the disc above and below. And these are cartilaginous, attached to bone, each about 0.6 to 1 millimeter thick, and they permit diffusion, and this is the main source of nutrition for the discs. The discs do not really have much of a vascularity. They get their nutrients and their water and their hydration from the end plates by diffusion. So disc physiology is that the discs also don't have much of a nerve supply. They are not very innervated except the very outer layers of the annulus. A healthy disc anyway. They also have minimal vascularity, as I said. They get their nutrients from the end plates. Blood vessels near the disc bone junction of the vertebral bodies, as well as those in the outer annulus, supply the necessary nutrients, as well as by diffusion. So a disc is a very strange part of the body. It's almost a protected place that doesn't have a lot of organic substance in terms of blood supply and living materials like nerve supply. So diffusion supplies this. It's almost like one of the reasons why discitis is so significant. Once you get an infection in the disc, it's hard to eradicate because there's very little blood supply. So let's switch gears for just a minute. Go back to the chronic inflammation in cells, and chronic inflammation is associated with not only disease but with aging, with loss of tensile stretching in tissues, loss of resilience, less flexibility, more brittle nature of orthopedic structures like the spine. Cellular senescence, or this aging of the cells, is associated also with the release of pro-inflammatory proteins. So as a cell is becoming inflamed and aging itself, then it sends out signals to its neighbors saying, this is an old folks home, we should all age. You should all act like this, like me. A cell affects its neighbors that way through signaling molecules, which we'll circle back to. Also, senescence of cells is a response to stress, physical stress, and nutritional stress, other types of stress. In fact, inflammatory networks during cellular senescence have been found to affect even remote tissues in the body. So disease begets disease. Inflammation begets inflammation, and aging begets aging of not only the cell but the organism. So what happens in a problematic or a painful disc is we have degradation of the extracellular matrix proteins, the ones I listed earlier for you, the proteoglycans, the glycoproteins, the elastins, collagens, etc. They don't turn over. They're not being produced well. They lose their viscoelastic or squishy property. They become dehydrated. Fewer proteins brings fewer water. You get sclerosis of the end plate, less diffusion, less nutrients. It's a problematic feedback loop. And the more vertebral end plate pressure, the more the marrow edema changes, and you get those modic type changes on the MRI that I mentioned to you. Now, physiologic or organismal age is a manifestation of sorts of the cellular age, but we really see the difference when we have plenty of years to look at it. So age of the organism can feel and look different than the actual temporal age, meaning the physiological age of something and the temporal age of something are not necessarily the same thing. Here I have a picture of two different hands, for example, and assuming they're the same temporal age, they clearly, one looks much drier and wrinkled, and you see changes in the joints and aged, if you will, compared to the younger hand. So how do we have a healthy, prolonged age? Well, first we start with the cells, so the organism can have that. Some people call this the health span. And we know some things. First of all, exceptional longevity runs in certain families. And in those families, in those genetics, the diseases that they do get are compressed towards the end of those exceptionally long lives. There is a genetic influence on survival. The older you get, the older you will likely stay or become or get. Meaning the older you get, the older you will live, oddly. Many disease-associated genetic variants are seen in elder populations and centenarians. The variants have something to do with the slowing of aging and the decreased risk of age-related disease. Now, age-related disease is a funny thing because the longer you live, the more likely you are to accumulate a disease. But in this case, the more accumulation of inflammation your cells have with age, the quicker you will age. In other words, the less inflammation, the more controlled, or the better proteins, the less urgent signaling that one inflamed cells does to others, the less of that, the slower the aging process or the healthy prolonged age one will have. Think of it at the cellular level. Inflammation also it can be from oxidation. Oxidation can be from environmental stresses and things like this. Oxidation is bad to our DNA, so you're more likely to be inflamed if you're oxidizing. So why is one person aging better than the other? And again, this is at the cellular level, and we're going to use this information to study the spine and apply it to the spine, because people have back pain. It's a huge effect upon populations, on loss of work hours, loss of productivity, use of pain medicines. If we can help neck and back pain, we've done a great deal. The spine on the left is a healthy spine. The discs are hydrated. The disc structures are not perturbed. The vertebral end plates have minimal marrow changes. I mean, that's a great looking spine. On the right, we have bone spur formation. We have disc herniations. We have shortened disc height. We have dry disc. We see modic changes in the bone. This is a very degenerated spine. This spine has a lot of inflammation, and this spine is a painful spine. So if we know how to affect the aging of that spine, we catch it early, we can affect the cells of that spine and help it from becoming painful. Just like many people start Botox injections early to prevent the wrinkles from forming, because once a wrinkle starts forming in the forehead, then the furrows get deeper and deeper and they're harder to help. But from cellular activity, we know from the healthy young stem cell signaling in vitro that these cells give out signals to their neighbors. These are called extracellular vesicles, also called exosomes. And I'm going to use the word exosomes more frequently here because it's the buzzword now. And these exosomes from a young, healthy stem cell can send signals, these exosome signals, to neighboring stem cells for them to turn on their apparatus and send out their exosomes and their messages, turn on their printing presses, communicate with their neighbors. Just like an amplified signal, like a meme goes out on the internet, and one friend sends it to three friends, and three send it to two or three each, and it amplifies. We know that these are akin to some of the important shock proteins, the heat shock proteins. These are in exosomes. We see miRNAs, which go into a cell and tell that cell what proteins to make without affecting its genetics in the nucleus. There's no DNA. And there are other growth factors in exosomes still to be elucidated. Exosomes from a healthy stem cell are different than exosomes from a disease cell or an inflamed cell. So that's why we're looking at therapeutics here and using healthy young stem cells to help aging and inflamed cells in someone with spine problems or with other diseases for that matter. So as I said, oxidation affects your DNA. Anything you do to protect your DNA would be helpful to slow inflammation. And there are supplements you can take. Caffeine actually is one of them. And there are molecular targets as well. But the clue to longevity is measuring the telomeres. And the more compact the DNA, the longer the organism lives. And this is because the DNA is more protected. So all of these things go together. And we see this through the signaling of the cells. If we can keep those cells with more compact DNA, we've improved the cellular health, the longevity of the cell, longevity of the organism, and have a better healthy lifespan or health span. So the same pathways affect telomere length that we mentioned earlier in the inflammation cascade. Remember, inflammatory measure in the cell is this NF kappa beta. So that has a role in telomere length. So the less inflammation on a chronic basis, the less DNA damage. You protect the DNA when you have less inflammation, and the cell does not age as quickly, and the organism does not age as quickly. Heat shock proteins are another one of these. Heat shock proteins can be found in organisms that are preserved all the way back to bacteria and across all of the five kingdom. One of the heat shock proteins, and these are found in exosomes of young healthy stem cells, is anti-apoptotic. That means that it promotes cellular life, and it promotes NF-kappa beta, which remember it helps the telomeres. And there's a lot going on in the cell. We're not going to cover every last thing, but heat shock proteins are crucial. And these are very basic, small, and preserved proteins. The number next to a heat shock protein is usually its molecular weight. For example, HSP 70, 90. We see these in all organisms. So where's the spinal DNA? Remember, there are very few cells and discs. It's in the bone marrow, correct. So if we're going to have a therapeutic target, knowing what stem cells can do for the healthy nature of the cells, the telomere preservation, we want to attack the bone marrow. If we have healthier bone marrow, we're going to have a healthier disc by diffusion. I mean, that's where the health of the disc comes from. So this is interesting because we can take exosomes that come from young healthy stem cells. We can take the heat shock proteins within them. We can take the whole thing and get it into the marrow adjacent to the end plate, and by improving the marrow health and reducing the chronic inflammation in the marrow health and the healing type processes of the cells, the disc will get healthier. And that's what we're working on here. But I don't want to forget the extracellular matrix. And this is the reason why surgery done in utero does not leave a scar of much. What is found in extracellular matrix, and there are products available right now, amniotic extracellular matrix, is that it's got all these great proteins, the same things we find in discs, collagens, fibronectins, proteoglycans, glycoamidoglycans, but it also contains extracellular vesicles, exosomes. So this is very interesting. So if you've ever seen a fetus delivered to a baby who has had spina bifida surgery in utero weeks before delivery, they come out with nary a scar. So there's something about this amniotic fluid in the extracellular matrix, or something going on that at its youngest state has the most optimal healing. This is the lesson that we've learned and applied to spines here in our practice. There are, if you study the contents of amniotic fluid, we have abundant youthful stem cells at its most youthful state. We have extracellular vesicles or exosomes. We have all these great proteins, tumor necrosis factor. I mean, this is the best biological fluid on the planet. So the goals of any regenerative therapeutic would be to find something like what's in the amniotic fluid that limits acute inflammation, prevents chronic inflammation, focuses on healing as opposed to ongoing smoldering cellular activity, limits the oxidative stress, preserves the DNA, and increases the cell lifespan, and maintains a happy cascade of cellular processes. In other words, the right amount of reduced inflammation. And this is through exosomes. We're very excited about exosomes here at ReCELLebrate. So this is taking active mesenchymal stem cells, and they produce exosomes. They produce these extracellular vesicles. They can be collected from the extracellular fluid, spun down, and by size separated. They signal neighboring stem cells and neighboring differentiated cells to behave properly, to heal, to limit the acute inflammation, to prevent chronic inflammation, and for those cells to send out their own signals to their neighbors. So it's a good positive feedback loop. And where else can we get exosomes besides stem cells? We can get them from PRP. Not a lot, but it's in PRP. You can get it from autologous stem cells. They're a little bit of a pain to harvest. And heterologous stem cell sources like amniotic fluid. They can also be separated out, and that's what we're focused on now. And this is a huge move in the business cycle. All the biotech companies are looking at this. And as I said, exosomes contains mRNAs. They contain heat shock proteins and small growth factors. And they contain there's a bilipid membrane, just like a small cell. They're about 30 to 50 nanometers, sometimes a little bigger. They can be they're probably about 1/500th of the volume of a cell, and each cell can produce hundreds of thousands of these. Without getting too far along the line, the best recipe for the spine using this knowledge is to use the best exosomes. So if you harvest bone from an elder person for their bone marrow for stem cells, you've got older stem cells. They're not as active. You got to really highly concentrate them. Bone marrow harvest is painful. I don't believe heavily in fat sources, because fat source for stem cells, those fat stem cells are somewhat dysmorphic. They're sending out signals to store fat. I'm not looking for that. I'm looking for the healthy, young, optimal, fresh amniotic sources, and those are the exosomes that are my favorite for use in a therapeutic setting. And we take these. Oh, I'm going too fast. We take these, and I think in the future we're going to have designer exosomes that are going to be more specific, content specific, function specific, tissue specific. But right now we're not allowed to manipulate them here in the United States whatsoever. So for the spine, we look at the source of the pain, and we see where the physiologic problem is, and then we go back to that degenerated disc and we apply the exosomes. So what we're doing is in discogenic pain, we are applying the exosomes into the vertebral bone. This is an intraosseous or in the bone injection, and we're seeing amazing things. This is not first reported by me. This is reported first by Patine, who is now, after the 3-year follow-up, showing rehydration of cells. I mean, this is remarkable. Rehydration of discs. And the better the disc works, the less pressure on the end plate. The end plate cellular activity improves because of the exosomal and stem cell activities. It's like that 3-year-old with the laceration on the knee. It heals quicker, better, faster than an older person can heal. This is from the Patine article, where it's a slightly different technique, but rehydration of the L4-5 disc. It's brighter than it used to be. When it's black on the left, it's almost white on the right. This is the first, in terms of, as far as I know, situation where that's been seen, and that was for an intradiscal injection. And again, I think we're going to get better and quicker healing of the vertebral bone, and this has been reported anecdotally by the French Dr. Hernigo, and who's been doing this and sharing it with his disciples as well. So my solution for discogenic spinal pain is intervertebral subchondral exosomes along the end plates, and this I think you'll be seeing everywhere in the next 5 to 10 years. This goes right into the bone. I put it through the pedicle under fluoroscopic guidance. We try to get near the end plates surrounding the disc, and we're doing a study now to look at the MRIs before and after. This also comes out of the literature for other joints besides the spine, knees, where the subchondral technique, the intraosseous technique, is showing results. And instead of doing PRP or stem cells, we're jumping ahead to the exosomes. We can do a highly concentrated amniotic exosome dose and get significant increase in stem cell activity of a joint and of the spine. So regenerative medicine is not just a future treatment, it's a future way to improve one's health and be preventative. And I hope that I've stimulated some questions. And I appreciate your time and listening and inviting me to participate today. You can reach me, please do reach out to me by LinkedIn. Let's become colleagues. Please, if you have any questions, go to our website and it'll click through to an email. Send me a message. I'd love to meet all you. I know we're spread out throughout the world, and appreciate this chance to share globally the exciting things we can all do in the field of regenerative medicine, in this case, in the spine. Thank you again for your time.